and it's my great honor to introduce our first speaker, Anna Janeva. Anna did her undergraduate studies in biochemistry at the University of Heidelberg, with a master's thesis at MBEL Genome Biology Unit in the No Lab, focusing on H3.3 mutants in neuronal differentiation. She's currently a senior PhD student at the Helmholtz Munich Institutes of Epigenetics and Stem Cells in the lab of Dr. Eva Hormanseder. And I'm really excited to hear her talk today on AI to AC, digital reprogramming decodes H3K27 acetyl as a novel barrier to cell fate changes. So Anna, over to you.
Thank you, Ben, for the kind introduction. I think there's not much left for me to say other than to dig straight into the science, which has recently been hot off the press, and we are very excited to share the highlights of our work with you. As you might already be familiar, during development, cell fates are established, and during this process, where the cells acquire a complex set of epigenetic traits, they establish a so-called cellular memory, which faithfully propagates across cell divisions and stabilizes the identity of the differentiating cells. And what was long thought to be a one-way street in developmental biology turned out to be a dogma that was shattered in the 1950s by the pioneering work of John Gurdon, who showed that by injecting a differentiated nucleus into an enucleated egg, one can reprogram cells to a totipotent zygote, which eventually develops into an embryo, giving rise to new cell types, and finally resulting in a cloned adult organism. And even though this technique has been around for many decades now, the mechanisms still remain poorly understood. However, from different reprogramming approaches and from the work of many, many labs, we know that in order for reprogramming to be successful, it requires chromatin and transcriptome reprogramming. So what does that mean? If we look at nuclear transfer reprogramming from an endoderm cell to an ectoderm cell, we would ideally hope that the chromatin landscape of the starting cell type reprograms to the corresponding chromatin landscape of the reprogrammed cell. And we would also expect that the transcriptome undergoes a shift from the starting cell type to the desired cell type. So that means that there needs to be activation and inactivation of transcripts, and this needs to happen in a correct manner for functionality of the achieved cell types. However, we know that reprogramming in different approaches is quite inefficient, and in particular, in the case of nuclear transfer, we know that only about 10% of the cloned embryos survive. And of the surviving embryos, we can detect many developmental abnormalities. And this can be in part explained by incomplete reprogramming of the transcriptome, which has been attributed to epigenetic features of the donor nucleus, which can be maintained onto the reprogrammed cell and can lead to incomplete transcriptional reprogramming. So in this case, this is something that is also known as epigenetic memory, and this can actually come in two flavors. So what has been widely reported and accepted by the field is that there is a memory of a repressive chromatin state in which genes that were not expressed or lowly expressed in the starting cell type fail to activate in the reprogrammed cell. And this is something that is known as off memory, while something that has been the focus of our group is the memory of an active transcriptional state. So when we look at genes that are specific to the starting cell type and persist their expression in the wrong cell types of the cloned embryo, this is something that we have termed on memory. So our group has shown that epigenetic memory of active transcriptional states is an important barrier to cell fate reprogramming, and we aim to understand the underlying mechanism. So in order to do that, we have actually asked the question whether genes that retain this active memory are barcoded by epigenetic features. So we perform nuclear transfer and transcriptome profiling in two different setups. First, from mesoderm to endoderm, and second, from endoderm to ectoderm. And we collected the tissues from the donor cells and from the reprogrammed cells. And what we like to use as a control for the normal transcriptome, we collect tissues from in vitro fertilized embryos. And then we also profiled a long list of chromatin marks, which were both active or repressive. And in order to make sense out of this huge body of data that we got and to identify whether there are certain hidden or complex patterns in these data sets, we developed a machine learning model based on convolutional neural networks, which could represent the chromatin and transcriptome data as images, and basically analyze which of the genes that demonstrate certain characteristics would be resistant to reprogramming or memory genes, or which genes would correctly reprogram or not fall into any of these categories. So with this, we developed digital reprogramming, which is a machine learning based tool which can identify novel epigenetic barriers to reprogramming and predict reprogramming outcomes on a transcriptome level. So in order to understand not only which could be the barriers, but also whether they whether these chromatin mark specific clusters of on-memory genes, we sort of use this tool known as DeepExplain that helped us calculate the contributions of each mark to the memory status of genes, simply put. And what we get as a result from this is such a heat map in which we can cluster the on-memory genes, and we can see which clusters have certain geo signatures and which mark contributed to the memory status of this cluster of on-memory genes. So in particular, we were interested to see that K4 trimethylation, which has been reported in the past, was confirmed with this approach, and we could see in particular in cluster 4 and 1, which are pertinent to endoderm development. This mark was particularly enriched, and in the case where we are looking from endoderm to ectoderm reprogramming, this hints that this mark could be linked to genes that are relevant for cell identities, and this could be genes that maintain memory. And then the next mark that actually popped up was K27 acetylation, for which we saw enrichment in cluster 1. Among others, but then when we performed such comparison also to correctly reprogrammed genes, indeed K27 acetyl showed the biggest difference. So that's how we chose this target to follow up on. And then we looked at the promoters and putative enhancers of on-memory genes in contrast to reprogrammed down genes. And similar to the previously published mark K4 trimethylation, we could observe strong enrichment on the regulatory elements of on-memory genes compared to correctly reprogrammed genes. So then we developed an experimental approach to perturb histone acetylation in donor embryos before nuclear transfer. So for this purpose, we treated the donor cells with P300 inhibitors to prevent the deposition of acetyl marks, profiled the embryos biochemically and genome-wide, and performed a nuclear transfer to understand how this perturbation in the donor nuclei affects the transcriptome outcome and the development of the cloned embryos. So just briefly, we used two different inhibitors of P300, one bromodomain inhibitor and one inhibitor of the catalytic core. And we could see, based on mass spectrometry analysis, that the bromodomain approach mostly reduced K27 acetyl levels, while the catalytic inhibitor showed a reduction in other histone marks as well. So with this, we sort of had two tools in our pocket, one which could be fairly selective for K27 acetyl, and one that showed a broader effect on histone acetylation. So then we went ahead and performed nuclear transfer with both of these perturbed donor nuclei, so with both inhibition approaches. And the way that we like to assess the outcome of this is using such MA plots in which we plot the expression of the genes in the donor nuclei, and on the y-axis we have the log-fold change between the expression in tissues of cloned embryos versus IVF embryos. So we could see that under control conditions, we could detect about 1,300 on and off memory genes. And then when we looked at the bromodomain inhibition condition, which is CBP30, we could see that the number of on-memory genes dropped to about 1,000, and we could observe that actually not only the numbers, but also the expression levels of on-memory genes dropped with this approach. And a similar trend was followed by the catalytic inhibition approach, in which we could see that there was a reduction in the on-memory phenotype, yet more modest than with the bromodomain inhibition approach. And importantly, when we checked the development of the cloned embryos, we could actually observe that after passing the gastrulation checkpoint in development, the nuclear transfer embryos that were derived from the perturbed acetylation conditions developed at higher rates, and we also observed healthier morphologies. So then we also wanted to check whether we can connect this improvement in the transcriptome and this modest improvement in development to candidate genes that could be rescued with the approaches. So we performed a stringent filtering of the on-memory genes that were rescued with the P300 inhibition approaches, and we could actually find that many endoderm marker genes could be successfully rescued in the nuclear transfer embryos, which means that they were no longer aberrantly expressed in the nuclear transfer tissues to an extent without the perturbation. So we can see that the expression in the donor nuclei was maintained, but in the NT embryos, this was reduced. And the genes that you can see marked in bold are known as endoderm marker genes. So we decided to call these rescue genes sensitive to the treatment because they became permissive to reprogramming. And then we decided to look whether we can actually link this to regulatory elements on which K27 acetyl is deposited. So as I mentioned in the beginning, this mark was enriched both on promoters and putative enhancers. So we decided to look at the genomic distribution of the mark upon perturbation. And we also decided to look at whether upon this perturbation, there was effects on other marks as well. So I will talk about K27 acetylation today, but you can ask me about preliminary data in the Q&A. So we wanted to see whether the proximity to an enhancer could be a feature of on-memory genes compared to reprogrammed genes. And indeed, we could detect that a higher proportion of on-memory genes was proximal to an enhancer compared to correctly reprogrammed genes. But did this also reflect on the genes that we could see sensitive to the treatment? And the answer is yes, because we could see that about 1/3 of the genes that were sensitive to the treatment were enriched for enhancers. So that prompted us to think that the P300 inhibition approach likely perturbed K27 acetylation on enhancer elements, which could have been behind the phenotypes observed. So we did cut and run in control and perturbed donor embryos, and we could see indeed that the majority of the genomic loci where the K27 acetyl levels were reduced were P300 sites outside of promoters, or in our case, these were sites that we could define as predicted enhancers. And then we stratified these genomic sites based on the proximity to gene sets, based on whether they were on-memory genes sensitive or insensitive to the treatment, or correctly reprogrammed genes, or neither of those. And we could actually see a significant reduction of K27 acetylation on enhancers that were proximal to sensitive genes. And this was also something that is nicely illustrated in this genome browser snapshots. Yeah, and this brings me to my final data slide for today. We also wanted to check whether we can detect any correlations between the changes in K27 acetylation versus the gene expression changes that we detected. So we did this first for the gene expression changes in the donor embryos, and we could not see, no, first, yeah. So in the donor embryos, we could see that there was a negative correlation between K27 acetyl changes and changes in gene expression, while in the nuclear transfer embryos, we could see that there was a positive correlation between changes of K27 acetyl on enhancers and RNA-seq log fold changes. So with this, I come to the summary, which is that in this study, we could develop a digital reprogramming approach, which based on a machine learning approach could predict reprogramming resistance from chromatin and transcriptome data. We uncovered K27 acetylation as a novel barrier to sulfate changes induced by nuclear transfer. And when we inhibited P300, we could see that this led to global K27 acetyl reduction, mainly on enhancers. And with this, we could observe that there was a loss of on-memory and improved developmental outcome of NT embryos. So last but not least, I would like to thank the lab for being great and always supportive. Chris, who developed the machine learning approach, and Sarah, who supported us with the enhancer prediction, and everyone else who supported us with this work. And for any questions, I'm happy to answer, and you can also read up on the recent preprint.
Some
Happy to
fantastic
take your
talk, Anna.
questions.
That was I really enjoyed that. For everyone in the audience, you can type your questions into the Q&A or click the raise hand button and I'll unmute you and allow you to ask your question yourself. For me to start off with, I thought this work is really interesting and there's lots of like potential questions to ask and like things to do from here. I was wondering, have you looked much at transcription factor expression with your P300 inhibitors? And so like one model that comes to mind is that maybe there's some key transcription factors that are kind of still being expressed when they shouldn't, and that's leading to this on-memory effect. And by inhibiting P300, you kind of break that cycle because a lot of P300 dependent genes, and you know, a lot of those are transcription factors. And so have you tried analyzing the data in that direction to see if there are like some number of key transcription factors that are P300 dependent and giving you some of these the better reprogramming?
Yeah, so indeed, the majority of the on-memory genes are transcription factors, and this also very obviously pops up when I try to do any kind of gene ontology on the on-memory genes. And also the genes that I was talking about that we could see that were sensitized up by the treatment and became permissive to reprogramming, they are also mostly transcription factors. So this is something that we could detect with the P300 inhibition in the donor nuclei, that we can sort of mitigate the on-memory of transcription factors that are markers of the starting cell lineage. And we even have data in a preprint that also recently came out from our lab that even when we knock down one of these transcription factors in the donor embryos, we can actually observe a strong rescue of phenotypes that were related to differentiation defects in certain cell types. So indeed, the transcription factors play a role, and their aberrant expression in the wrong cell types is something that could be driving this, we believe. Yeah.
Excellent. And then there's a question, there's a bunch of questions now coming in the Q&A, and I'll just ask one and then you can respond to the others by typing. But this question is, can you perturb memory at individual loci by modifying like a dead Cas9 type approach? And is that something you're thinking about trying?
Yeah, this is something that we haven't been that we haven't done yet, but it's in the works. So this will be very exciting for us to find out.
Awesome. Well, thank you so much. That's all the time we have. All the time we have for questions. Like I said, there's a few more in the Q&As. You can type your answers there. But
I
thank
will.
you again for a
Yeah.
fantastic talk.
Thank you.